Okay, good morning and welcome to the first of two talks this week on NMR spectroscopy. On Thursday, Jane Wagstaff is going to show some non-routine research collaborations that illustrate the context of the type of work that we're doing for biomolecular NMR in LMB. And today I'm going to set this up by taking a look at what is biomolecular NMR. We'll introduce it from the point of view of somebody who has never come across the technique at all. My preference is that we keep it very mathematically light. So we're going to focus on sort of cartoon illustrations to illustrate the principles of what's going on. And what we will look at is the basis of the technique. Where does the NMR signal come from? We'll look at what properties of a molecule influence the NMR spectrum, and so therefore we can work back from and record an NMR spectrum and infer from it what the biophysical properties of a molecule are. And then we'll take a sort of wider overview look at the broad scope of what NMR is capable of doing. Then later in the week, Jane will look at some more specific examples of exactly how you apply it in our sort of context. Now, anywhere that you get a movement of charged particles, you have associated with that a local magnetic field. And we're very familiar with this in the idea of electromagnetic induction and currents passing around electrical circuits. But the same is true at the level of the molecule, that the individual charged subatomic particles, the movement of those particles are responsible for generating a local magnetic landscape within the molecule that is a characteristic of that individual molecule. NMR is a technique that uses this landscape of local internal fields within a molecule to give information about the composition, the three-dimensional shape, and the way in which that three-dimensional shape is changing in time, the dynamic properties of the molecule. Okay, so that's the basic idea of what biomolecular NMR is all about. If you want to use NMR in your own research programs within LMB, your first point of contact is to go to the internal webpage, go to the intranet, look through the Scientific Facilities tab, and you will find details of who are the points of contact, who can you go to first of all to discuss details about what you want to do. You are not expected to be an expert in NMR from the outset. It is very detailed in the way in which data is acquired and analyzed in the end, and it requires a fair bit of experience. If you have that experience, you have the equipment available to you to go ahead. If you don't have the experience, contact the NMR facility, talk about what you want. They'll help you design the program, implement it, and then look at the data analysis afterwards. It's what the NMR facility people, the NMR facilities support staff are there for. It's collaborating on projects within the LMB. Physically, we're not located in the same building as the rest of LMB. We're about 5 minutes walk away, nice and socially distanced out in a place that is it's near the blood bank on the Addenbrooke's Hospital site. And one of the reasons for putting all of the equipment there is because we then don't get any interference from other equipment, from other scientific techniques. And also our stray magnetic fields don't impinge on any of the biophysical techniques that are occurring within the LMB building. So, I should say that the dreaded N word is prohibited effectively on a clinical site. Because we have neighbours and we have residential blocks around, it's just not necessary to alert people by using the word nuclear in the magnetic resonance. So we reduce the term from nuclear magnetic resonance to magnetic resonance spectroscopy. This is in contrast to the magnetic resonance imaging facilities that are used in other parts of the hospital in the clinical setting. Okay. So we're usually referred to as MRS on a clinical site rather than NMR. The facilities that we have are exceedingly good. They're state-of-the-art facilities. We have at the moment 6 spectrometers. We've lost one this year. We've retired one that was originally put in in the late 1980s, and finally we retired it this year. But we have different spectrometers operating from 400 megahertz that is used to support the organic chemistry efforts through 5, 6, 7, and 800 megahertz. And we also have access to state-of-the-art 950 megahertz facilities. We're one of the major users, external users of the Francis Crick Institute NMR Centre in London. So that's basically, all of these machines are configured for observing proteins, labeled proteins that are labeled with the stable isotopes C13 and M15. And we also have on each of our machines here, except the 400s, we have this system of pumping through cryogenically cooled gaseous helium. So the detector coils are actually at about roughly 20 Kelvin. The sample itself is held at room temperature, but the electronics are cooled in this, what's referred to as a cryoprobe, to take out the noise from the system. And all of our machines are equipped with these high sensitivity cryoprobe detectors and optimally configured for looking at biological samples. So what's the purpose of the investment? What do we expect to do with the investment in that kind of equipment? Well, the primary application of NMR spectroscopy is to look at the composition of molecules, and pretty much every major university in the world now has some access to NMR spectroscopy for the characterization of its organic chemistry, organic and inorganic chemistry compounds, its material stuff. So that's it's ubiquitous throughout the university and commercial system sectors. The other major applications, excuse me, in the context of biomolecular NMR is to look at solution structure. Now we can do this either to look at de novo structure determination, where we use NMR data on its own to produce a folded protein structure that is consistent with the data. Or we can take a structure that is generated by some other AlphaFold or similar technique and use the NMR data to validate whether this is in the correct state or not. And we can then look at, if we've got an overall structural fold for this, we can look at, for example, open and closed states of particular switching mechanisms. So we get to look at both the overall structure of the molecule and how it's changing with time, whether it's in open and closed states. Remember that proteins aren't static, rigid structures. The dynamics of a protein are very much instrumental in the mode of action and the biological functioning. We can look at residual structure in intrinsically disordered proteins. We have a principal advantage of being able to look at the association of molecules. So NMR is very sensitive at identifying the interfaces and the presence of binding between two different components. And we can characterize those in some minute detail by performing data-driven docking simulations using NMR data to dock a ligand onto a protein surface and get a model of the ligand-bound states. We have a principal advantage that NMR provides over other forms of spectroscopy, is that we see an individual signal for every individual atomic nucleus in the molecule. So we can work out at site-specific detail around the molecule what's going on at any one time. And I'll come back to this again and again because it's a crucial advantage of what we can provide by NMR that we don't believe that you can provide with other techniques in many cases. We can look at the stability of the protein in terms of its hydrogen exchange kinetics. We can look at HD exchange on a per residue basis, solvent exchange, that is, of exchangeable hydrogens. We can also measure the radius of gyration, radius of hydration, and diffusion constants for the molecule. NMR is a quantitative technique, so it enables us to pull out details of biophysical constants. We can measure kinetic rate constants, equilibrium constants, pKa's, whatever. It is a quantitative technique. The pharmaceutical industry has adopted NMR as one of the principal screening techniques for lead generation. And NMR provides a suite of techniques that allow you to identify small molecules and fragment molecules that bind to targets. We can characterize binding affinities, at least rank order those binding affinities, and identify what does, what doesn't bind, and where it binds on the protein surface. Does it hit the active site? We shouldn't be too fixated on looking at the macromolecule itself, though. Biomolecular NMR can encompass so much more. And we can also get a lot by direct observation, either of the substrate or of the small molecules itself, and get involved even in metabolic studies and looking at metabolites in whole cells. So there's a broad overview of the type of work that we can do. So we can map the binding interfaces, we can identify the locus of changes, and we can look at structural perturbations and post-translational modifications. The sort of context that I've been doing a lot of my work on in the last few years is looking at a complex like this. It's a multi-protein assembly that is the focus of interest of Marianne Bientz's research group in LMB. It's a transcriptional activation complex called the Wnt enhanceosome. And rather than looking at the entirety of this complex as it is assembled, we can look at binary components and look at specifically the interaction, how they interact. We get a lot of domain swapped homodimers forming in this case, and we can look at these sorts of processes with NMR and really get a mechanistic principle on how a complex like this assembles. So we can look at the direct binding interactions. We can characterize what are the binding surfaces, the contact surfaces between them. We can look at the complexes, whether they form stable or very transiently populated complexes. And it's important for us to stress that we do this as an integrated structural biology approach in combination with the other biophysical techniques that you're hearing about in these series, and particularly the structural techniques of cryo-EM and crystallography. So NMR isn't a panacea. There are obviously things it can't do. And one of the bits where we get into a bit of difficulty sometimes is where we have planes of symmetry. So if you look, for example, at the Buckminster fullerene C60 carbon structures, actually symmetry was one of the key features of being able to solve what the structure of this was in the first place, because for 60 carbons, you get one single peak in the NMR spectrum, which says all of these carbons have the same magnetic environment. There is a perfect symmetry for all 60. So in that case, actually symmetry works in favor of the NMR because it was able to identify that was the form of symmetry that's present. In this example from the Wnt signalosome components, we're looking at a homodimer that is domain swapped. So part of the structure from one subunit swaps over onto the other, and we have a plane of symmetry down the middle of that molecule. It's rather difficult for us by NMR to resolve that plane of symmetry and look at the two subunits individually. There is a way of doing it if we introduce carbon labeling into one of those units only, but we'll maybe come on to that later. There are some interesting examples we have of being able to get into cracking that kind of problem. NMR suffers a bit if you have heterogeneity. We start to get too many signals and it complicates the process. So in preference, if we can, we are looking for extremely pure monodisperse and preferably single conformers if we can. Monodisperse, it can be dimeric or trimeric, but as long as it's not a mixture of the monomer dimer trimer, we're much more happy as to detail we can pull out in this. We have the requirement of a minimum, absolute minimum of 280 microliters. There we go. Sorry. Just got a pop-up appeared on mine, and now it's gone. 280 microliters, and the minimum concentration that we can look at realistically, it's got to be upwards of 20 micromolar, which is quite a lot of sample compared with many biological applications. There is a fixed concentration for the sample, fixed volume for the sample, therefore the concentration becomes quite critical, and it always is in biological studies. Once the molecule starts to rotate, physically rotate in space, slower and slower, the quality of the signal that we get back from the molecule is degrading. So NMR suffers from going to high molecular weight. And it is often the case that we apply the technique to single domains rather than to multi-domain assemblies. NMR is much more suited to looking at single domains in isolation. Where you get a low spin density, particularly in nucleic acids, NMR suffers again because there aren't enough reporters of what the state of the system is in the middle. So we will require systems in which we have many coupled NMR active atomic nuclei. Nucleic acids are quite difficult, certainly not impossible, but proteins are somewhat easier with the higher spin density. And it's worth saying that the speed of analysis, speed of data analysis, certainly for structural work in NMR, can be relatively slow in comparison to crystallography. If you want to know the three-dimensional structure of a protein, on every occasion you would recommend going for crystallization if it ever will crystallize. The speed of data analysis, very often the same day, and even if not, after a couple of days of data analysis from crystallography, once you've got the data, then you have a structure at the end. But with NMR, it can take weeks or even months to polish off a publication quality, high resolution structure. So we would always recommend, if you can, you do your structural work by crystallography, but NMR can get in and get some quick answers on some of the other information. We have to be aware that NMR sees all of the states that are present in the solution. So each of the individual states contributes to the signal that we see back. And what we will see in the end is an overall average, ensemble average of what's happening in the sample. If we were to interpret this data in terms of a single structure, it's absolutely meaningless. It's what we would refer to as a virtual confirmation. You can get a picture that that's what the sample looks like, and it's not ever actually physically populated in this state. It has the contribution from the individual states in there. So we have to be very cautious whenever we're interpreting NMR data to consider that there are multiple confirmations. We cannot resolve individual confirmations very often, but what we can do is use NMR in combination with computational techniques to infer back what the individual contributing states in the system are. We can determine by NMR what is the extent of the dynamics that's occurring in the solution and what the rate is for the interconversion of those species. So NMR can give you information in that, but you have to be cautious always when you're interpreting NMR data of the ensemble average that's intrinsic. This is a slide that I've nicked from Jane from her talk later in the week, and it's just really to mesh in with what she's going to be saying. So we've decided that for fitting all of this stuff into the time slot that we have, Jane will look at the dynamics in more detail, and I will just scoot over it very briefly. So this is really just to introduce more of what she'll be saying later in the week. There are a whole variety of processes occurring on different timescales within a molecule. And NMR has a variety of techniques. This is not an exhaustive list, but it's a set of examples of procedures that we can use to probe the motions that are occurring at different timescales. And that's something I'm going to skip over today. and Jane will go on to next week. So let's look at where the NMR signal comes from. There are a collection of isotopes, stable isotopes that are not decaying, not undergoing radioactive decay. All of these isotopes have one single property, that they have a nuclear magnetic dipole. Okay, the ones that are in blue here are present at approximately 100% natural abundance. So if we look at any molecule that we take in solution, virtually all of the hydrogen will be in the H1 isotope. We use the term hydrogen and proton interchangeably in NMR because H1 nucleus is just a proton on its own. It's one proton with no neutrons, and that is the dominant isotope of hydrogen. So you very often use the term proton and hydrogen in NMR interchangeably. Phosphorus is also, 31P, is also a spin-1/2 nucleus that's there at 100% natural abundance. By spin-1/2, I mean it's got a nuclear magnetic dipole, a north and a south pole. Okay, and when we place that atomic nucleus in a magnetic field, it adopts one of two possible spin states. The quantum mechanical selection rules dictate that it can only adopt one of two individual spin states. And the energy of those two states is very different as we put the molecule into an external magnetic field, which we're going to designate with the term B0. So the overall construction of an NMR spectrometer is that we've got a cryomagnet here with high magnetic field, and we've got an external B0 field that it's applying to the sample. The sample sits right in the center of this magnet can. Okay, so as we increase the magnetic field, the energy difference between the two states, effectively with the north and south pole either aligned with or against the field, we are looking at an energy difference that is directly proportional to the B0 field. It has this proportionality constant gamma, which hides all sorts of the most complicated aspects of the nuclear physics that goes beneath it. It's all conveniently packaged into the single property called gamma, which is characteristic of a particular isotope. So hydrogen and phosphorus are available for free to us. We can also bring other nuclei into play, but they're there at low natural abundance. So if we want to use carbon or nitrogen in our NMR studies, we're going to have to explicitly put those isotopes in. So we bacterially express the proteins in a growth medium that contains either it contains C13 glucose and N15 ammonium chloride as the sole source of carbon and nitrogen. And in that way, we can incorporate NMR active isotopes of carbon and nitrogen into the proteins that we're trying to go for. I should say we can also do some labeling with deuterium, but that's used not to see something that you can pick up by NMR. That tends to be a technique for silencing the signals from the proton NMR. So we can incorporate deuterium to silence stuff. It's also worth saying there are no NMR usable, NMR friendly isotopes of oxygen or sulfur. And a lot of the ions that you come across, for example, zinc, there isn't a convenient NMR active nucleus. And so we can use surrogates. So directly below zinc in the periodic table is cadmium, and cadmium can be used as a surrogate of zinc binding to give us an NMR friendly and NMR active form of a metal that coordinates in pretty much the same way that cadmium does. We can look at lithium. We can look at fluorine in some cases. It's being used a lot in the pharmaceutical industry for screening projects. If you've got something like sodium that you wanted to see, you can use a surrogate such as rubidium, which is also a monovalent cation that you can use as a surrogate for sodium, but is more NMR friendly in our case. Okay, so it is worth saying that in the absence of any other magnetic field other than B0, all of the nuclei of a given isotope will resonate at exactly the same frequency, not near enough or thereabouts, but exactly the same frequency. So when we're talking about the field strength of these spectrometers, and very often you'll hear people talking about the higher field strength, what is the advantage? What are the relative merits of going to higher and higher B0 field strengths? Well, the first is you get a sensitivity increase. The signal to noise is proportional to the power of 3/2 of the B0 field. So we don't just increase by a linear contribution, we actually do slightly better than linear as we go to higher magnetic field strengths. So the magnetic field strength strictly here is in Tesla, but what we quote for the spectrometer is the frequency that you get for a proton nucleus at that field strength. So the spectrometers are always referred to by megahertz of the proton frequency. We also have an increased dispersion of signals. So at 400 megahertz, the signals spread out over this kind of absolute frequency range in hertz. That doubles, the spread of those frequencies doubles as we go to 800 megahertz. So you get a linear relationship between the signal dispersion and the magnetic field strength. You can see in some of these small distributions in here, they're just not resolved at 400, whereas they are resolved as you go to high field strength. So sheer resolving power of a spectrometer improves as you go to higher field strength at the disadvantage of cost. So roughly speaking, the cost of an NMR machine goes up by a factor of 2 for every 100 megahertz increase. So at the moment, for the top field strength that's available commercially, we're looking at 1.2 gigahertz, and the cost of a spectrometer comes in over £20 million. So that's at the top end. For something around the 400 end, we're talking about £300,000-400,000, something like that, for a start into the lower end of the field range. But the basic premise is at the end, the higher the field strength, the better, and you buy what you can afford and what's needed for the scientific projects. It brings us to a very interesting point, though, that if the absolute frequency changes as we change the field strength, then we need a scale, a frequency scale that is normalized against the strength of the spectrometer. So this is plotting exactly the same data as we've seen on the previous slide, except we're plotting it as a so-called chemical shift. Now, the chemical shift is the frequency normalized against magnetic field strength. So it's normalized against the absolute frequency of a reference spectrum. You tend to get that the differences in frequency that we see across our spectrum are about a millionth of the overall absolute frequency. And so we tend to introduce this term and multiply it by 10 to the 6 and just use the units ppm. So this is a ratio. The chemical shift value is a ratio of two frequencies, so it's unitless, dimensionless. But we tend to divide it by 10 to the 6 and use the units term, parts per million, to describe the frequency scale. But notice on all of these signals, this is looking at sucrose from the sugar bowl in our office, and the frequency, center frequency of all of these signals is the same, but we get better resolving power of these as we go to higher field strengths. That's what the chemical shift is all about. This is the business end of the spectrometer. It's the bit you never see, the bit buried in the middle of the magnet can. And we have a detector coil referred to as a probe, and it's sitting right in the center of the external magnetic field B0. A sample is put in a 5mm outer diameter tube, glass tube, and the sample never comes into contact with the machine itself, so we don't get contamination from the machine into the sample or the other way around. You can get your sample back at the end, providing it hasn't gone off, it hasn't degraded with time. So the sample solution, we're looking at an overall volume of 550 microliters in this tube, gives us the 4 centimeters depth that we need. So 550 microliters, and the concentration range is from 20 to usually 500 micromolar. We wouldn't normally recommend people going to higher than 500 micromolar routinely, because you tend to get the risk of aggregation and all sorts of artifacts that come through from aggregation of proteins. So we're looking in a good concentration range where we can get individual monomeric species in solution in that concentration range. The temperature for the sample itself is also good for biological studies. We can go down to 0 and up to about 60 degrees in the equipment that we've got. So the basic idea is all of the spins sit in the middle of, there we go, in the middle of this detector coil, and the coil is set up to detect any magnetization components that are rotating, that are precessing around this axis in the XY plane. So assuming that the Z dimension in this case is the coordinate axis, Z is straight up the bore of the magnet, and we're looking at magnetization that is in the XY plane. So we can prepare the magnetization, put it into an excited state where it is rotating in the XY plane, and in that configuration, the coil will pick up the rotation of these bulk magnetization vectors that are coming from the atomic nuclei in the sample. And what it generates for us is a sinusoidally oscillating signal that is oscillating at the frequency of rotation of the spins. This is the basic NMR signal. I should say that strictly speaking, this is the magnetization of the X component is described by a cosine wave in this particular case. But since I've written the term signal here, it's worth saying that the signal itself would also contain some contribution from anything that is in the orthogonal direction, in the Y direction as well. So strictly speaking, the term for the time dependence of the signal would be described by this, but we're approximating it at the moment as a signal that is oscillating with a cosine frequency. If that is an infinitely long cosine, we can transform that using the process of Fourier transformation, which is not a complicated process, very straightforward. We can transform that into a spectrum, a frequency spectrum, and extract the frequency components from it. And if this is an infinitely long cosine wave, what we get at the end is a delta function, a single, I'm missing one label from there, a single discrete frequency that is describing the frequency of that wave. In practice, we don't have an infinitely long signal. What we have is a signal that we put into the excited state, and then it rings down with an exponential decay. So we have an exponentially damped cosine wave. And when we do a Fourier transform on that, we get what's referred to as a Lorentzian line, which has not only the same frequency, same center frequency as we saw before, but it also has associated with it an amplitude and a line width at half height. So there is an uncertainty to the frequency that is expressed here as a line width. Okay. We should say that this line width is inversely proportional to the time constant for the exponential decay. So as the signal decays quicker, the line width goes up. And this is one of the reasons why NMR has a molecular weight dependence, why we start to get degradation of signal as we go to higher molecular weight. Because as we go to high molecular weight, the efficiency of relaxation back to the equilibrium state increases. So for high molecular weight systems, large proteins, they relax extremely efficiently back to equilibrium. The lifetime of the signal is very short, and the line width goes up at the end. For small molecules, what we'll see is there's a long duration for that signal. It decays very slowly, and we can get high resolution information with very narrow line widths for the signal. So that's what we're seeing in the next slide. The overall line width that we see for a macromolecule that's efficiently relaxing as it's tumbling slower and slower in solution. what we see is an efficiently relaxed signal, and it gives you very broad lines. Eventually, those lines will become so broad that the spectrum becomes unusable. For small molecules, we get the opposite. We get a very much longer lived signal, and it transforms to a sharper line in the spectrum. Now, I'm very impressed with the simple illustration, simple analogy that's used by Andy Derome in his book. And he envisages the NMR spectrum as analogous to the oscillation of a simple harmonic oscillator. And what he takes for this is a bell. Now, we're going to treat this as a bell with a single harmonic frequency, which is not how bells are. We're going to ignore the fact they have overtones and mixed vibrations. This is an analogy of a simple harmonic oscillator with a single frequency. And what we do is we ring the bell, we record the damped signal that rings down from that. We put it through the process of Fourier transformation and extract the frequency component of the oscillator. If we put another oscillator into the solution, the signal that we'll see at the end is actually a summation of these two lines. And if we put that through a Fourier transform, we'll extract both frequency components. And we can keep putting more oscillators into our box. And what we get is a cacophony from those. And as we do the Fourier transform process, we can extract the individual frequency components of the original oscillators. So that is directly the analogy for an NMR spectrum. What we're going to do is analogous to taking the box and shaking it up, we're going to excite all of these oscillators simultaneously with white light in the radio frequency range. They're all stimulated at the same time. We record the cacophony that comes off in the detector coil at the end, run it through the process of Fourier transformation, and extract the frequency spectrum for the components that are in there. So when we look at part of the proton NMR spectrum of a 10 kilodalton protein, this gives us some illustration of the immediate difficulty that we're going to run into. There's just so many signals in there that there's a limited amount of information that we can take due to the amount of signal overlap that we see inside the spectrum. So what we're going to need to do is simplify the spectrum by spreading it out into another dimension. So this is referred to as two-dimensional NMR. It is actually the same spectrum that we see here, but in this case, we spread the signals out in a second dimension according to the nitrogen frequency, the spin frequency of the directly bonded nitrogen. So in other words, we've transferred some magnetization through the bonding network from a proton to its directly bonded nitrogen, and we've got a two-dimensional plot now of the frequency of the proton signal versus separated by the frequency of its directly bonded M15. We don't have time to go into at the moment exactly how we do the transfer process, but if you want to know more details, you can always come and see us afterwards. But the basic take-home principle from this is that we have a two-dimensional map now. Although that may seem more complicated, it's actually what it's done is it's given us a simplified spectrum by spreading the signals out more and giving us better resolution. This isn't a particularly useful representation to look at the data. It's a bit cumbersome. So always you will see NMR spectra plotted as a contour plot where we look directly down. Again, this is the same data set. We're just looking at different representations of the same data. And we're now looking down on those peaks from above, and the intensity is depicted as contours. So what's going to be quite useful is if we look at what are the basic principles that dictate what is in an NMR spectrum and how what it contains. And where I'm going to start is might look at first like it's quite complicated. It's the nuclear spin Hamiltonian describing the energy of a spin in a magnetic field, but I don't put that in there to make it look complicated. I put it in for exactly the opposite reason, to make it look very, very simple, because in fact, it's just a summation. Everything we see in NMR is a summation of three separate terms. The first is referred to as shielding, and it gives us the intrinsic frequency of the signal. the so-called chemical shift that we talked about already. We got another term called J coupling and a third term called dipolar coupling. And what I'll do is just briefly touch on what these are, because they are the three components that when we sum them up, they make everything that happens inside the NMR spectrometer. So the first of these is shielding. And the concept is that around every atomic nucleus, NMR active atomic nucleus with this nuclear dipole, magnetic dipole, we have a circulating cloud of electrons, and those electrons that circulate directly around the nucleus always operate in generating their local magnetic fields that opposes the applied magnetic field. Always these, the valence electrons will be generating a magnetic field that opposes. So we talk about this idea of shielding, where the nucleus is shielded from the external magnetic field by this local field generated by the circulating electrons. So that's what we're referring to as shielding. What it means is that the frequency that we see, the effective field that is seen at the nucleus, is obviously dictates what its spin frequency is going to be. And we can infer a huge amount of detail about the electronic structure, the electron density that is around each of the nuclei. So by far the dominant feature of the NMR spectrum is the functional group chemistry, the local composition of each of the centers. Remember, we get a separate signal for every nucleus that is in the molecule. So we get structural compositional information at the level of functional group chemistry out for every nucleus in the molecule. So it gives us a characteristic frequency, and as we said before, we depict this as normalized against the field strength. So in every spectrometer in the world, it will come out with the same chemical shift value for the rotation, the spin speed of that nucleus. So this is one of the things I said in the first slide, a landscape of local magnetic fields that is unique to every molecule that we put in the spectrometer. And so it's this landscape of magnetic fields that is so useful in giving us information about the functional group chemistry, the composition, but also the three-dimensional structure of what's going on. We have other magnetic currents that occur locally. So this is from the passage and movement of delocalized electrons. So for example, in aromatic rings, you get a pi cloud with rotation and circulation of electrons that generates profound magnetic fields. And if you put an aromatic ring from a phenylalanine or tyrosine anywhere near any other group of a protein, it has a profound effect on the chemical shift values that you see in all of the amino acids in the vicinity. And the same goes for circulation of electrons around other pi bonded systems, including carbonyl groups, where you get areas in which the magnetic field is either enhanced or suppressed by the effect of the delocalized circulation of electrons. The second effect that we wanted to look at was J coupling. This is an effect that is transmitted through covalent bonds. So actually the effect is transmitted between by the magnetic field from the covalently shared electrons. And so what we have is the frequency or the environments that we see this spin in can either be enhanced or it can be opposed, slightly shielded by the effect of the magnetic fields in the covalent bonds. And it depends upon the spin state of the neighboring nucleus. So we get two spin states where the nuclear dipole is effectively opposed or with or opposed to the field. And it gives us two different environments at the site of this spin in gray here. So in fact, we don't see a single frequency signal. What we see is that that signal is split into two, and the splitting of that is a constant value that is dependent upon the field that is generated by these covalently bonded electrons, and it's referred to as the J coupling constant. So in fact, rather than seeing one signal, we see two, and it's split by J. The magnitude of J tells us information about the strength of the contact, the efficiency of the contact through those bonds. So it depends on the geometry, the torsion angles that you have around the atomic bonds and the extent of all the orbital overlap that you get in the covalent bonds in the network. So it gives us some information about the geometry by measuring the magnitude of J coupling constants. The final term, the third of these three terms, is perhaps more intuitive, is that if we're looking at the magnetic environment of this spin in grey, it's going to be influenced by the magnetic dipoles generated by other nuclei that are in the environment. And this works through space rather than through atomic bonds, and it works up to a distance of about roughly 7 angstroms away. So we're talking about 4.5 carbon-carbon single bond lengths. That sort of distance that we can use these or notice the influence of other spins. And it's the direct influence of whether this spin is in a state, the neighboring spin is in a state where its magnetic dipole is with the field or opposed to the field. So in fact, although most of the time we talk about this separation being J, it is in fact the J coupling constant plus the D, the dipolar coupling, the influence of the field that comes from dipolar coupling. Dipolar coupling has a profound influence on NMR spectra, and it has both a distance dependence that we've just seen, but it also has an angular dependence, and the angle that this bond makes with the external magnetic field has an influence on what we measure for the dipolar coupling, strength of the dipolar coupling. And for molecules that we put in solution, they're tumbling very rapidly, and this splitting term averages over all possible orientations and all possible angles of theta here. So the average splitting that we see for a molecule that is sitting in solution, tumbling isotropically, is 0. So you don't see any splitting in the spectrum from the dipolar coupling. It doesn't mean that dipolar coupling disappears. It's just that the average splitting that we get for the dipolar coupling is 0. If we don't exchange all of those different bond orientations, so the sample basically is a solid state sample and it's sitting there, we resolve all possible orientations of this angle theta, and what we get is a spectrum here in blue. So this is the sort of width of spectrum of signal dispersion that we would see for the chemical shifts of a typical proton spectrum. If we include a solid state spectrum for that, and this includes the dipolar coupling terms, then dipolar coupling dominates over everything, and we get a loss of detailed information in there, and the spectrum is of somewhat little use at this point because we've lost any fine detail in the spectrum. So when you're looking at solid state samples, you are taking, the whole basis of the technique is to remove the dipolar coupling by instrumental means that I'm not going to go into because it's not something that we do in-house. We don't look at solid state samples. It can certainly be done, and there are constant progress in the solid state NMR of proteins. It's not something that we do in-house at LMB, but we do have some collaborations that have gone on with solid state NMR groups who do some of this kind of work. But that's not something that we'll cover today. One of the things where I say the dipolar coupling term doesn't disappear, and it is with us in the form of this process referred to as nuclear Overhauser enhancement, or the nuclear Overhauser effect, or NOE. The nuclear Overhauser effect is effectively an interatomic ruler, an internuclear ruler. And the strength of this effect is directly proportional to the inverse 6th power of the distance. Okay, so it gives us the ability, we can measure the strength of this NOE effect, which is just basically a difference, a non-equilibrium population of electron energy levels, of magnetic energy levels. So it's an energy level distribution, but just referred to as the NOE. And if we can measure the strength of the NOE, we have a direct measure of internuclear distance between two specific spins. But we can also take an alternative approach, particularly if we were to look at the dipolar coupling between two spins that are covalently bonded to each other. So for example, an H and an N in the background of an amide group in the backbone amide of a protein. What we would see is that distance term now is fixed, it doesn't vary. And if we look at any differences in the magnitude of dipolar coupling, it is giving us a measure of the bond angle. So it gives us an angular term. So if we were to take a sample and put it in a magnetic field where we don't get completely isotropic tumbling, what we can see is we would get a slight preferred orientation of the protein with its axis of diffusion slightly aligned with respect to the magnetic field. What you would see at that point is that we're introducing a non-averaging, non-uniform averaging of this angular term, and you would have a residual dipolar coupling term that is dependent upon the orientation of this bond vector with respect to the major diffusion axis of the molecule. Okay, so we have two different terms that we can pull out with dipolar coupling. One is the NOE, which is a measure of internuclear distance. The other is this residual dipolar coupling if we put the sample in a medium that gives it non-uniform motional averaging. Okay, if we look at the growth of protein structure by NMR in solution, it really grew up from the early 1980s when Kurt Wuttrich's group in Zurich were using NOE-based approaches to determine protein structure. And the idea is that inside the protein, there are thousands of protons. We take a frequency assignment of each of those spins. So it's a laborious process, and we get an assigned frequency for every one of those spins in the molecule. We can then measure the strength of NOE between each of the pairs of these that are within 7 angstroms of each other. We put those into some kind of distance geometry, minimization, and you can get the overall fold of the protein. So this NOE-based structural analysis began in Kurt Wuttrich's group in the 1980s. So the structure is determined from short-range HH interactions, and we apply something upwards of 25 NOE restraints for each amino acid residue in there. Somewhere between 25 and 40 is typical. This is the basic NMR spectrum, the 2D NMR spectrum that you would use for the NOEs. And it is referred to as a nosy. What is showing us in this particular case, we can take one of these peaks here. It's telling us that a peak that is at that frequency is close in space to a spin that is at that frequency. So we can read off the coordinates of the two. Because we have the assignments for the frequencies of all of the spins in there, we can tell exactly what pair of spins this is referring to, and the strength of that interaction is proportional to the strength of the NOE that's in the solution. So what we're seeing is the stronger the signal here, the closer two spins are. So we can measure from the intensity of that peak how close two spins are and exactly which two spins it's referring to. In this spectrum, we got something like 7,000 peaks, and the traditional process of interpretation of this is that you have to get in there and interpret them by hand. Certainly, it's one of those processes that is in active research interest for the artificial intelligence community, looking at ways of automating this. But at the moment, as things stand, we don't have reliable enough automated interpretation of these data. So you get in there and you interpret this data by hand. As we go up in molecular weights, the quality of these spectra degrades because the line widths go up. So a realistic molecular weight limit for doing a high-risk NMR solution structure is something like 40 kilodaltons. Certainly there have been some in Lewis K's group going up to 86 kilodaltons. That's an absolutely non-routine example. The realistic limit is somewhere about 30, 40 kilodaltons for a high resolution structure of a protein. We can also apply the residual dipolar couplings. The idea in this case is that we record a proton spectrum for a protein that is tumbling isotropically in solution. So we just dissolve the protein up, record its proton spectrum, and what we see is a separation of this signal. It splits into two according to the coupling constant between that proton and its directly bonded nitrogen. And in this case, we can measure that. That is the value of J, as we said in the earlier slide. Now what we can do is take the same proton and we put it in the presence of a weak alignment medium. So this is a medium that has collimated structure inside the solvent. And what it gives us is non-uniform averaging of the orientation of the protein. And now we have a residual dipolar coupling. So we can measure the separation of these signals. That is the value of J plus the residual dipolar coupling. This isotropic spectrum is the reference spectrum, which is just the J value on its own, and the dipolar coupling is averaged to 0 in this case. So we can extract from that what the value of the dipolar coupling is. So this occurs where we've got a sample where we have 1 part in 10 to the 3 excess population of its major diffusion axis aligned with the B0 field. And we've managed to recover this few hertz of residual dipolar coupling. That may look absolutely underwhelming when we see it as a diagram like this. That's the process. When we look at a real example, very trivial example in this case, it's a small 3-helix bundle. And what you notice in there is that the NH bond, the axis of that bond orientation, is roughly collinear with the helix axis, very roughly. And when we look at the magnitude of the RDCs that we measure, residue in that backbone for the NHs, what we see is we get 3 regions of RDCs in there, and they correlate very well with the orientation of the helices. And in fact, we can do a lot to refine that structure and tell in some high resolution what the angular difference is between each of these helices in a bundle. That for a 3-helix bundle is just illustrating the point, but in practice we can use it for proteins of a very much more challenging application. This is P53, full-length P53 as a model. It's a homotetramer, and each of the 4 subunits in here, we've modeled on the N-terminal domain, which is an intrinsically disordered region. Residues 1 to 93 for the N-terminal domain are intrinsically disordered. We put 20 copies of this N-terminal domain onto each of the 4 subunits on here. And we've measured RDCs, several RDCs in this case, not just the NH RDCs. We also did the C alpha, C beta, and we did a CO to N, and we get a unique orientation for each peptide plane that is in that molecule. And when we take the experimental data in blue here, and we simulate it against a random coil, a true random coil, the data don't fit. Right? The simulated data don't fit with the experimental. In fact, the simulated data only fit if we introduce 2 turns of alpha helix in this position and about 22% polyproline helix towards the N-terminal end of this. And what this does, this is showing that we've got a stiffening of the chain towards the N-terminal. So in fact, that helps to project out this N-terminal domain, this intrinsically disordered segment, from the body of the protein. And this happens to coincide with one of the known recognition sites for binding partners of p53. So this is one application in which you can use intrinsic RDCs to look at residual structure in intrinsically disordered proteins. We can also use chemical shifts themselves. Now, chemical shifts are screaming information at us about the whole state of the magnetic environment around each of the signals that's in the nucleus, that's in the protein. So what we can do in this case, we can construct something that's referred to as a random coil index. And this is just calculated from the measured backbone chemical shifts and how they differ from what you see in a true random coil. So we use it, the random coil index is built up from 6 different chemical shift values from the different nuclei. And it can be applied either to folded proteins, as in this case, just illustrating what's going on, or it could also be applied to look at residual structure in intrinsically disordered regions as well. It gives us the propensity, the probability, if you like, of forming secondary structural elements throughout the protein. And what I've done is I've colored this structure with the color bars that we get from the random coil index. And you can see that we've got 4 strands of beta sheet in here, and we've got 3 helices, and these are very well in accord with what we measure from the NMR data. So this information we get for free just from measuring the chemical shift assignments in the first place. And all of this is implemented within the program Talos N, which is a software package that's freely available from the NIH from Adbax's group. It also includes this thing called an order parameter, which describes the rigidity of the chain. So we can see loops in this particular case. Okay, the last technique I won't say very much about is similar to putting a FRET label into a fluorescence resonance energy transfer label into a molecule. We can put a paramagnetic spin label into a protein, and it will enhance the relaxation rate, and we can measure that in the spectrum, and it will give us the idea of the distance between a spin and the paramagnetic label based on how much we enhance the relaxation rate of the protein. So what we do is we have a paramagnetic species that induces rapid relaxation in any spin that's within 35 angstroms. And then we can reduce this from the paramagnetic nitroxide radical into the hydroxylamine, which is diamagnetic and doesn't relax neighboring spins. So we record the spectrum in the before and after the addition of the scorbate, and it gives us an idea of how much the spins are affected and how close they are to the label. This can be used either intramolecularly or intermolecularly, and it can be used to look at detecting stable or transiently populated states. So we've got a whole load of NMR data that can be used as information giving on the three-dimensional state of a protein. And we use the traditional methods of molecular mechanics calculations, in which we take point masses on Hooke's law-like springs for every bond angle, bond length that's in the molecule. That's sort of intrinsic. But if you were to run those kind of algorithms without the addition of NMR restraints, you normally get absolute rubbish out at the end. So you can improve the accuracy of these kinds of molecular mechanics models by adding in restraints from the NMR data. And that is the NOE values, these pairwise HH distances, the residual dipolar coupling, the chemical shift index, the residual chemical shift, the random coil index that we saw, J coupling constants, hydrogen bonds that we can infer from hydrogen bond exchange rates, and these PRE spin labels that we put in. We add all of the information that we get in and supplement this force field with terms from the experimental data to get final solution structure. The overall procedure, the most common way of doing it is to look at simulated annealing. So what we have is an energy landscape. Now I illustrate it for one coordinate, but obviously inside a protein, we have thousands of different coordinates that need to be optimized simultaneously for all the different bond angles and bond lengths. If we were to take a minimization procedure on a reaction coordinate or a conformational coordinate like that, and we just apply minimization, we may go into what is not the global overall minimum for the structure. So the way that we can increase confidence that we found the global minimum is we give the system enough energy to traverse all of these energy barriers, and then we slowly take out energy from the system, thermal energy as a simulation from the system. And slowly this system should converge down to the lowest energy point as we get down to zero kinetic energy, zero thermal energy. So how can you be sure that you've reached the global minimum? Well, the best way of doing that is that you run multiple simulations and you start 100 different starting points along this coordinate. And if they all converge to the same point and they are consistent with the experimental data, then you have a greater confidence that this represents the true solution structure. But this is what I want to pull out from this slide, is it means that when you deposit an NMR structure into one of the structural data banks, what you have is an ensemble overlay of multiple states from this simulated annealing run. When you deposit a crystal structure, you deposit a photograph of a single conformation of a crystal. Right? In NMR solution structures that you download from the data banks, it is downloaded as a solution ensemble. Nowadays, we are exceedingly excited by the development of the artificial intelligence algorithms, and particularly AlphaFold and what they're doing for protein structure. NMR is going to be one of the huge beneficiaries of AlphaFold, because the slowest step in NMR solution structure generation was doing de novo solution structures in the first place. So what we have here is in pink, I've got the latest structure that we did that just came out last month of a domain with 114 residues that we did by conventional NOE-based NMR approaches. And we overlay it with the AlphaFold structure that it shows exceedingly good correlation between the two structures, but we also, in the context of AlphaFold, get a wider context of that. So this AlphaFold structure obviously takes a lot less time to generate, and we can then use the NMR data to validate the model that we get out from AlphaFold. We also have other guided homology modeling routines, CS Rosetta. CS stands for chemical shifts, so it uses chemical shifts as a restraint to guide a homology-based modeling of protein structure, and it's producing very high reliability models very rapidly. So the whole procedure of structure generation is revolutionizing within NMR, and we can then validate these structures by checking them and see which conformational state they're in, and look at conformational switches by using a limited set of NOEs rather than going through the thousands and thousands of NOE assignments from before. We can use limited sets of dipolar couplings and the comparison with random coil chemical shifts. Characteristics of the protein NMR spectrum, the proton NMR spectrum, and basically it's dominated, the regions of the spectrum, it's dominated by the electronic environment, so the composition. So the HN all resonate in one region, H alphas in a separate region, and any methyl groups in the side chains come in a separate region altogether. The archetypal spectrum that we use, though, is referred to as an HSQC, and we can only get this for a protein if we introduce N15 as a stable label into the protein. So we bacterially express in a medium that contains N15 ammonium chloride as the sole nitrogen source. What it gives us is a set of spectra, or a spectrum with a set of peaks that is one backbone amide for every amino acid residue in the molecule. And we get a separate reporter group for what's happening at 200 individual positions throughout that protein. So we can start introducing binding partners, and if we get anything that binds anywhere close, you'll see some of the resonances that are nowhere near the binding site just are completely unaffected by the addition of a binding partner, but we can map out what the magnitude of the changes are on those resonances that are near the binding site. And at that point, we can use fitting algorithms even to do this as plot this out as the perturbation that we get in the chemical shift in the HSQC as a function of ligand concentration. And from that, we can get accurate information on affinities. This is quite a trivial example on the previous one, but here's something that's more, a little bit more challenging. It comes from some work from Stefan Freund from a few years ago. And what he's done is color-coded the structural changes that you get, the changes that you can infer from chemical shift perturbation as you add a binding partner. This is looking at blood clotting factor, thrombin, and it's binding to 3 different ligands. And you can do all combinations of the chemical shift assignments to work out what is the structural consequence of binding each of these individual ligands in place. We can look at high resolution images of dot structures. So if we do a filtered nosy experiment, in this one, for example, we are rejecting any signal of a proton that is bonded to a C13 in this dimension. So in other words, we only pick up the signal from protons that are C12 bound in this axis, and we only allow C13 bound proton signals in the horizontal axis. Might seem confusing at first, but what it gives us basically is a set of NOEs that are going from an unlabeled ligand that doesn't have C13 in it onto a C13 labeled protein. So all of the NOEs that you see here are going across the interface between the unlabeled ligand and the labeled protein. I've color-coded all of the chemical shift perturbation that we see when we add the ligand, and that's the sort of shift perturbation. And when we add in the measured restraints from 35 intermolecular NOEs that we see across the interface here, what we can do is a flexible body docking routine that puts this ligand into the protein site. We're asked, what's the molecular weight for a molecular weight limit for a protein? Well, there isn't really a limit. It depends what information you want. As we said, for high resolution structures, you're looking at about 40 kilodaltons. But even if you've got a larger complex, we can still tell if when you add a binding partner, you've got perturbation of the spectrum. And there are things that we can tell about how significant that is for the functionality of the protein. So there isn't really a hard and fast limit. It just depends on what information, what degree of resolution do you need to get from the data. We shouldn't be fixated on looking at the protein itself. This is an example from Jan Lerver's group where we're looking at the turnover of ATP, and it gives us some idea of the functional integrity of the enzyme under different conditions. And we follow that not by looking at the enzyme, which in this case was a huge assembly, too big to do by NMR, but we can just simply look at the hydrolysis of ATP. We can also look at metabolomics, and this is looking at intact erythrocytes. Now, if we take a spectrum of a whole intact erythrocyte, you see a broad background for the macromolecules, very broad signals for the slowly tumbling macromolecules, and superimposed on that envelope are sharp signals for the small molecule metabolites. Surely it couldn't be so simple that if we were to just delay our start of data acquisition for a few milliseconds at the beginning, we could remove all of the signal from the background of the macromolecules. But in fact, that's exactly what happens. We just introduce what's called a relaxation filter. And from that, we can extract out the high molecular weight components or filter out the high molecular weight components. And what we're left with is high resolution structures of the small molecule metabolites. And there are groups out there that are characterizing in real time the progression of hundreds of biomarkers simultaneously from the same spectrum. We can use NMR as a coarse grain NMR, mass spectrometer. It is not a mass spectrometer, but it certainly gives us enough resolution to say whether we've got monomer, dimer, trimer. Okay, so you can do this with a so-called 1D spin echo and other related experiments that allow us to get within a few kilodaltons what the effective molecular weight based on its tumbling time and how rapidly it tumbles in solution. That's something that we can do as a quality control check on your protein. Is your protein folded or unfolded? Well, this is a typical profile in the NH region of a protein that's unfolded. All of the NHs in the background are similarly solvated in similar environments, and so they all come up with roughly the same resonance frequency. If we put this into a folded protein, they spread out and disperse over a wider range. This is instantly characteristic of whether your protein is folded or unfolded, so it gives you a quality control check. Similarly, on the other end, we've got methyl groups that are all in the same environment in an unfolded protein, but they disperse out in a folded protein. So we can look at an HSQC for a normal folded protein. This is what we get for an unfolded protein. It's very much less dispersed, so we can instantly see that this is unfolded. I'm being photobombed by a cat. And we also have a characteristic appearance of a spectrum that is aggregated for an aggregated protein. This is extremely useful when we're doing NMR assays because we can continuously check a quick HSQC on a sample and look to see if it's changed since we first put it in. A lot of assays are being done out there on proteins that have gone off and proteins that are in aggregated or unfolded states. When you're doing the assays by NMR, you can see the quality of the protein and you can see exactly when the protein has gone off. It's an extremely important part of the work that we do. Okay, you can encode dynamic behavior into the intensity of an HSQC. We could even do HD exchange. So we dissolve this protein in deuterium oxide as a solvent. And these signals will start to exchange away their NH signals, so that exchange with solvent. And we can plot the signal intensity as a function of time and get the exchange rate. Or we can use it to look at dynamic properties, and that's what Jane is going to look at in her talk in a couple of days' time. So we've got this whole range of experiments that can be used to program into the intensity of this peak. Note then that we have the reporter group at each of these different sites. We know which spot corresponds to which amino acid, and therefore we've got dynamic content encoded into this HSQC data with per residue resolution. That we can illustrate with this real example from Alan Warren's work. Alan Warren is a clinician with interest in oncology. He's looking at this particular protein, which is 3 domains together. And you can see that 2 of the domains, these 2, rotate as a single rigid body, and the 3rd domain rotates independently of the other 2. So it has a degree of flexibility in the linker. Mutations at these points are associated with clinical outcomes, with pathology in the clinic. And what we can also see in this particular case is we're comparing human and yeast protein. And you can see that this beta-6 strand in this position in the yeast protein is not folded with the rest of the protein. In other words, this beta-6 strand comes out and is independently rotating, much more flexible than the rest. There are not many techniques that can pull out that kind of detail that Stefan Freund has got here from the backbone dynamics of this protein. So I'm very aware, I've gone way over time and I do apologize for that. We can look at site-specific detail of all sorts of exchange processes. So this can be individual pKa values, it can be post-translational modification, phosphorylation, folding kinetics or binding. Importantly, we can follow the change on a site-specific basis and extract biophysical constants and rate constants for different enzymatic turnovers. We are able to use NMR in fragment screening. Every pharmaceutical company has used fragment screening, and NMR is one of the bedrocks of doing that for lead generation. I'm not going to go through this now, there's no time left, but it's a collection of different experiments that we can use, either observing the NMR spectrum of the ligand, the small molecules in libraries, chemical libraries, or we can observe the signal from the protein in order to get proof of binding and generate, do lead development. And different techniques in there. There's the overall summary slide that is showing that we're looking at the protein QC, we're looking at structure and dynamics, we're looking at intermolecular interactions and the quantification of rate constants and exchange processes. Some suggested reading in there, just a few bits of a glossary, and that's the acknowledgement slide. We've used examples from various people that we collaborated with at the bottom. And thank you for listening. Jane will be in on Friday, and if anybody has any questions that they want to ask, I'll be certainly very pleased to answer those. Okay, if there's no questions, then I will close that off, and Jane will be along on Thursday.